Sta partendo. Ok, prego. Prof, scusi, non la sentiamo bene, non la sentiamo bene. No, prof, non si sente bene l'audio. Fino a 3 minuti fa sì.
Il problema. Ora mi sentite?
Eccoci, eccoci, sì, prego.
Ok, allora, buongiorno a tutti, abbiamo il piacere di avere con noi il professor...
Scusi, prof, si può fare tutto direttamente anche la presentazione in inglese, partiamo tutto quanto in inglese.
Sì, scusate.
Ci sono anche studenti esterni, prego.
This is morning science, is not scienza del giovedì mattina. So we have to speak in English, and it's nice that there are people from other languages that can follow our seminars also from online. So we have the pleasure to have here the Professor Massimiliano Aschi. Massimiliano Aschi is a professor of the University of L'Aquila. Is a full professor of general chemistry. Massimiliano Aschi graduated in chemistry, and he did a PhD also in chemistry in the University of Rome Sapienza. And he was later postdoctoral fellow in Berlin, in Perugia, then in Viterbo and in Tor Vergata. And since 2001, I guess, he is a staff member of this university as associate professor in general chemistry and then full professor in general chemistry. During his career, he was always devoted to the study of computational chemistry and tried to apply this field to these techniques, to develop the techniques of computational chemistry and try to apply also the techniques to different fields, such as biomolecules, physical chemistry, and also pharmaceutical chemistry. So please, Massimiliano, the floor is yours, and go ahead.
Okay, thank you, Leonardo. Thanks to organizers. I'm very happy to be here. Today, it was my intention to give you a very brief description of our activity. I will not go into the details, chemical details, mathematical details, and physical details. So it's a very qualitative seminar. Of course, all the people who might be interested in go more into the details of what I'm going to tell you is welcome. You can contact me and I can provide you with any kind of support, of course, literature and so on. I switched the camera in order to not make the connection too problematic, if it's fine for you. Do you see the shared screen?
Yes.
Okay. So, okay. This is the title, and you see that there is something apparently strange because computational chemistries, because as you will see in a moment, in my opinion, it doesn't exist only one computational chemistry. And in particular, the connection with applied science. In other words, since I'm talking to people who are not involved in computational chemistry, and most of you are involved in biosciences, so from our point of view, applied sciences, I thought to organize, to arrange my talk just to see how could we connect this very strange discipline, I mean very obscure discipline in some cases, which is computational chemistry, with more real sciences, I mean sciences who are dealing with real problems, how to say. Okay, this is the outline of my talk. I hope that I will be fast enough. In any case, you can stop me whenever you wish. So I will start with this very, very brief review of this computational chemistry. What kind of computational chemistries we can locate in the literature and in the science. What could be the connection between computational chemistry and other applied disciplines? And finally, I will give you a brief example of my topic, which is actually also the topic of Leonardo Guidoni, which is the computational physical chemistry, providing you with a very simple example just to give you a flavor of what can be done. And in particular, this example will also show you what are the limitations of computational chemistry from your point of view. So, first of all, I would like to introduce you to computational science in general. Computational science is a sort of science which is in the overlap of different disciplines. Mathematical disciplines, which provide us with algorithms and provide us with the language of science. Technical sciences, like informatics, software development, hardware development, graphical development, and so on. And most importantly for me, scientific disciplines, physics, first of all, then chemistry, engineering, biology, medicine, also economics, architecture, and so on. Of course, computational chemistry is only a part of computational science. In particular, is a part of computational science which makes use, in particular, of mainly physics, chemistry, and biology. In other words, sciences which are more interested to fundamental arguments. And we can say that the role of computational chemistry is the interpretation and prediction of biological, chemical, and physical processes, but looking all these processes at atomic and molecular scale. In other words, we can make a lot of computational science in biology, chemistry, and physics, but this science becomes computational chemistry when our point of view is from the atomistic point of view, from the molecular point of view, not beyond. Okay, now, in my opinion, there are two main branches of computational chemistry. Of course, this subdivision may be somewhat, subjective, and which is the basis of a very long discussion. It's not my intention to do the discussion here. So it's a very, very rough subdivision. On the left side, you see what I call bioinformatics or artificial intelligence. And on the right side, I have defined what I call computational physical chemistry. What are the differences between two kinds of disciplines? On the left side, you see that we have a huge number of tools essentially based on very complex algorithms, based mainly on statistics, but relatively simple and, in my opinion, sometimes completely absent, physical models. In other words, bioinformatics and artificial intelligence has the aim of predicting physical, chemical, and biological phenomena without being very interested in the physical inside, so in the knowledge of what is actually happening in what we are looking at. On the other hand, computational physical chemistry makes use of a lot of technical methods, molecular mechanics, molecular dynamics, Monte Carlo methods, quantum chemistry, and so on, quantum mechanics, in which the roles, as you see, are exactly inverted. In other words, we have relatively simple algorithms, but very complex or very complex physical models. It means that with computational physical chemistry, we may reach a very high physical insight in the phenomena, so we can understand what is actually happening, but as you will see in a moment, we do not have a very large predictive capability. So it means that if you want to predict something, you may be more interested in the left side of this panel. If you want to understand, you are more interested in the left side of this panel. Of course, these two sides of the panels in the last years are merging together, but the route, in my opinion, is still very long. For better understanding what I have just said, I would like to show you a very simple, very trivial example. Most of you know what is the flow chart of drug discovery. In the left side, you see from the top to the bottom all the steps required for arriving at a drug. Well, computational chemistry is more or less in that position. So more or less in the position in which we need to understand or to predict what is the ability of this potential drug in making a very effective biological action. If you consider bioinformatics, or more in general, artificial intelligence, the way we are going to follow is to try a huge number of possible candidates. You see, you can arrive very easily to millions or even more candidates, and to see how these candidates more or less in a very poor physical scenario interact with our receptor. Of course, this requires, as I told you just a few minutes ago, a very complex treatment of a huge number of data, but on the other side, we have a very simple physical model. In other words, for example, in most of these cases, we do not consider the solvent, that's the medium in which most of these processes, basically all these processes take place, and the receptor is typically treated as a rigid scaffold. On the other hand, if you want to find a drug following computational physical chemistry, we have to follow a very different route. In other words, we select one molecule, for example, a molecule whose activity is very well assessed, and we try to understand all the details of the interaction of this molecule with the receptor, treating all the system in a very physically coherent way. Of course, what is better of these two methods? Of course, none of the methods is the best one, because if you want a very fast result with a very high probability of success, you have to follow bioinformatics or artificial intelligence. On the other hand, if you want to understand how the drug behaves, it's better to follow computational physical chemistry. If you have a look at the literature in molecular biology, cell biology, medicine, physiology, medicinal chemistry, in other words, all the disciplines which we can easily define, very roughly define, applied science, you see that nowadays the ratio is clear. I mean, most of the people are more interested in artificial intelligence. In other words, in a very fast result with a relatively high probability of success. But unfortunately for you, I work on the other side. So I work in computational physical chemistry, which is not very well followed by people not working in our area. And so I will try to show you why. I will try to I want to start with a very brief review of what we can do today with computational chemistry. In this space, bidimensional space, you see on the y-axis the energy involved in the process, and in the x-axis the complexity of the system, which might be roughly connected to the number of particles. You see that in the high energy, very low number of particles, we have the single atom, the single molecule, or very, very ordinated system like the crystals. In other words, nowadays we can study with very large details these systems with quantum mechanics. This is the part of computational physical chemistry which deals with electronic structure theory, both studying the mechanical features and also the dynamical features. On the other hand, in the right part, the low part, we have phenomena which take place with a very lower amount of energy. In other words, no chemical reactions are taking place and are treated with classical mechanics. So the atoms motion is treated with classical mechanics, and all the particles which should be described by quantum mechanics are not taken into account. So it's what we call classical molecular mechanics and molecular dynamics. This means that there is a region of this space, which more or less is here, that is very, very difficult to address. And this region, unfortunately, is the region more interesting for chemists and for biologists. IDEST is the region in which there are chemical reactions or spectroscopical phenomena in complex environment, for example, in solution, or within a cell, or within a protein, and so on. And our work is more or less located in this region. So we are we try to do a very difficult stuff, but probably I will show you in a moment what we can do now. Okay, to give you a flavor of what we can do in this part of the space, I want to tell you very briefly our point of view for the enzyme reactions, which is a topic typically residing in this part of this space, because for studying enzyme reactions we need both quantum mechanics, because in the chemical reactions the electrons move, but we also need classical mechanics, because we are dealing with very, very large systems in which quantum mechanics cannot be applied for technical reasons, and so we have to use classical mechanics. All of you know what an enzyme does. An enzyme is a sort of catalyst. In other words, an enzyme is able to lower the free energy barrier of a reaction, leaving the thermodynamics of the reaction, which is in the right part of this figure, unaltered. So the thermodynamics is not affected, is only affected the kinetics of the process. But what does such a picture really mean? This is our question. This is the question that we are doing to ourselves for many, many years. For trying to give you an answer to this very complex question, I want to show you a very simple example. This example is taken from an old work of our group, more or less 15 years ago, in which we have tried to investigate the electron transfer reaction in the copper zinc superoxide dismutase. As most of you know, the reaction, the basic reaction in which this enzyme is involved is the transformation of a radical anion into, in particular, oxygen radical anion in water peroxide. This reaction is very important by a physiological point of view because almost 20 years ago, it was discovered that a simple mutation in a region very fast from the active site, I forgot to remark, is indicated by the arrow. So a single mutation very far from the active site makes this enzyme essentially unable to promote the first step of the reaction. In other words, a point mutation very far from the active site makes this enzyme unable of reducing the copper and so oxidizing the oxygen radical anion. It's important to remark that the crystal structure of the native protein, so the wild type, and the mutant protein is exactly the same. We have been told in many years that the protein structure is essential for the protein activity. So this claim seems to be in contradiction with what we have learned on the textbook. So what does it mean? What's happening? We are losing something important? In other words, how is it possible that an apparently very insignificant mutation, very far from the active site, which doesn't produce a significant change in structure, is able to inactivate this protein? To answer this question, which is more or less the same question that we have seen a few minutes ago, but more detailed question, we have to repeat, it's important to repeat, what is the action of the enzyme. When we have the mutation, in other words, when we use the killer mutation, we are in this situation. In other words, the free energy is so high that the electron is unable to go from the copper, from the oxygen to the copper. When we remove the mutation, so when we go back to the wild type enzyme, the barrier is lower and the electron can easily go from the oxygen to the copper. The question is, how does the enzyme lower the free energy barrier? This is the question. So what is the action of an enzyme on the free energy? I want to stress that in chemistry, we use to explain the chemical reaction not in mechanical terms. In other words, the molecules and atoms do move according to the laws of the mechanics, but in chemistry we do not use these arguments, but on the other hand, we use more thermodynamics arguments, I would say more statistical arguments, like, for example, free energy. And this is in other words, the connection between the world of single atoms and the world of the number of Avogadro of atoms. So now we have to address this question, this very important question. To answer this question, we have to consider a further question. And the further question is the blue one. What do the active site and the substrate feel inside the protein and in the presence of the solvent at the physiological temperature? This is not a trivial question, but it's very important. Well, the substrate inside the protein, in the presence of the solvent, feels an electric field. So the protein, the enzyme, is a generator of electric field. A generator of the electric field on the active site. But the other question is, this electric field is static or is dynamic? And of course, to answer this question, we have to consider that both the protein and the solvent can be static or dynamic. And all we know that the protein and the solvent are dynamical systems. So when they are in a physiological condition at a finite temperature, so they are not at 0 Kelvin, they move, so they fluctuate, so they are dynamical objects. And this means that the electric field that they produce on the substrate is a dynamical object. In other words, the active site feels a fluctuating electric field. So the electric field felt by the place in which the reaction is taking place is fluctuating, is not static. And we have demonstrated that this fluctuation of the electric field is the key, is the way in which the enzyme lowers the free energy barrier. In other words, we can perceive the enzyme as a perfect nanomachine, which is optimized not only in terms of three-dimensional structure, but is optimized also in terms of mechanical behavior. I mean, it's really a nanomachine. It's not a nanostatic object. It's a nanomachine. In other words, it's a nanodynamical object. And this is very important because we can learn from the nature not only the structural argument, but also the mechanical argument. Now, the final question, which makes understand more in detail the role of the enzyme, is why this polyphenoation is not able of lowering the barrier. The question is probably, sorry, the answer is probably trivial at this point of the talk. Because if the effect of the protein is produced by the fluctuating electric field, the point mutation induces a change in the electric field fluctuation, which makes the protein unable of lowering the free energy barrier. And in fact, we were able to evaluate within the protein, inside the protein, so on the active side, the distribution of the electric field produced by the protein and by the solvent. And you see that this distribution is different. It means that a single mutation produces a different movement of the protein, so a different, how to say, a different electric field which the protein is able of producing on the active side. Now, what I've been showing you up to now is really exploitable for applied science. The answer is that probably not, because as I told you, in applied science, this kind of investigation is not very well considered, at least in quantitative terms. Why? Because in applied science, we need to address real systems and not simplified models. Most of you work in cell biology. So when we apply the computational physical chemistry in a cell problem, can we remove the rest of the cell and focus our attention on a sub-portion? And of course, this is not very, I think, probably not. And why? The problem is that we should consider the whole cellular environment, but this is not possible because the chemical reaction of the electron transfer takes place in a very low time scale, but the motion of the enzyme takes place in a very long time scale. And larger is the system, longer is the time scale, and much more complicated are the calculations. So the problem in considering very detailed physico-chemical interpretation of the phenomena, the problem is that when the system is becoming too large in terms of time scale of the phenomenon, the physical-chemical tools become not longer applicable, at least nowadays. So what would physical chemistry, what would make computational physical chemistry really attractive for applied science? Of course, the possibility of investigating very different time scales from the single electron transfer to the huge motion of the whole biological environment. For doing that, we need a lot of things. In particular, we need a very rapid development of computational results, very rapid adjustment of algorithm efficiency. In other words, we need more efficient machines, computers, more efficient algorithms, and most importantly, we need to consider that we still want to understand, and we do not only want to predict the phenomenon. So we need to maintain the scientific curiosity to understand the single event taking place in a very complex phenomenon. So I hope that it was fast enough, clear enough, and that's it. If you have questions, I'm.
Okay, I think one question is already in this line. So why people this is more psychology than science, but it's interesting. Why people are more interested in predicting instead of understanding a specific phenomenon nowadays? Question.
How should I answer? I mean, when we write projects, we claim to save human lives. And I'm joking, of course, but people is more interested in predicting because, of course, most of the scientists are moving to applied problems. For example, pharmaceutical chemistry or biochemistry or medicine. And in these cases, it's of course more important to be able to predict something than to understand something. In other words, again, going back to the superoxide dismutase, nowadays is probably more important to discover a drug able of preventing this infection rather than to understand why this infection comes out, in my opinion, of course. I don't know if you agree or...
You are muted.
I can't hear you.
Sorry, I have a bit of lag from my clicking. So now I think you can hear me. So
Yes.
I'd like just to add some comments about your presentation. I liked the example that you have you showed. And in particular, I like the fact that, I mean, the role of dynamics and fluctuations, which is something which is not clear directly from the static crystal structure, the picture of the book, no? The book picture. This is something that is it's actually present in many other examples, and it's very interesting, I think, to show to people that are working in biology.
Yes, absolutely. Yes, I would say that it's present. Is an ubiquitous phenomenon. It's present in all the biological systems, in my opinion. In some cases, it's more important. In other cases, probably it's less important. We have demonstrated that it's very important, for example, in electron transfer processes. It's a bit less important in, for example, proton transfer processes. And there is a reason for that, but it's not, I have not time to discuss this point, of course. Probably it's not interesting for most of the people present, I guess.
Okay, then there is a question from Michela. So I don't know if people can actually ask question live, or should I read the chat? What is the policy of?
It's fine. I can ask by myself.
Okay.
Okay. So thank you, Professor. It's really nice.
I can't hear you. I can't hear you.
Sorry. Can you hear me now?
Yes.
Okay. So it's a really nice talk and really interesting. My curiosity, I'm not a chemist, I'm a biologist, so I can go in the details. But given the complementary of the computer physical chemistry and the artificial intelligence that you told at the very beginning, is there a way to make them work together?
Yes.
Okay.
Yes, there is a way. I did mention this at the very beginning. It's becoming in the last years. It's becoming very diffuse. The attempt of using artificial intelligence with understanding the complexity of, for example, a protein fluctuation derived from computational physical chemistry tool. In particular, when a protein moves, the most difficult part of the interpretation of this motion is just to understand how is taking place. It's like the vibration of a molecule. Understanding the vibration of a molecule is not trivial, because we observe an object in a Cartesian space, but if you look at this object in Cartesian space, you completely lose the physical insight and the physical meaning of his movement in biological terms, I mean. So, for example, when the motion of a loop, it's taking place in the Cartesian space, but the biologist claims that the loop is moving. So how the biologist can say that the loop is moving is he or she is making a transformation from the Cartesian coordinate to a sort of internal coordinates, which is the loop. And this is the most difficult part of the work. And many people is trying to solve this problem using artificial intelligence. Of course, I think that there are still many years to come for arriving at a very efficient interplay between these two apparently separated disciplines.
Thank you so much.
Thank you. Yeah, this I think it's important. I had the same question actually, so it's good that someone else also asked. And there is another question from Anna Teti.
Yeah.
Maybe you can.
Yes.
Sure.
Hi.
Ciao, Anna.
Yes. Ciao. So I don't switch on the camera because the connection is unstable. But I have a couple of questions. One is, do you know if this particular mutation can give rise to a genetic disease? Are there, because the enzyme is an important enzyme, so
Yes.
you know if this is true also for diseases. So
in the case of SOD, it is a genetic disease. Yes, it is.
so with this specific mutation, you mean?
Yes, this specific. It was found from people who was affected by Goering disease. Goering disease. I don't know.
Oh, okay. Okay. Yeah.
And this was a very peculiar question going around in the literature, and we tried to address this question. Of course, I don't want to claim that our interpretation is the reality, but I only want to claim that we observed a sort of realistic interconnection between the different mechanical behavior and the different chemical behavior of the wild type and the mutant. That's it.
Okay, this is good. So you were not playing around. You were doing something really very important.
Yeah.
Another question I have is more general. Are there situations of mutations in amino acid that are far from the active site that modify the molecule in a different way, for instance, making them sensitive to pH changes or binding some cofactors like calcium or other?
Yes, the answer is yes, definitely. Yes, you can. For example, we have been studying some proteins, membrane proteins, whose action is affected by a mutation not in the membrane but outside the membrane. So this means that this change is transferred from a very far portion of the protein to the place in which, for example, the chemical event takes place. For example, as you told, this might be an amino acid mutation or even a change in the pK of an amino acid, changing, for example, the local ionic strength of something else. It's a very complicated story, but it's very fascinating at the same time.
No, I found this type of research really fascinating. First of all, I understood what you were saying, and I'm not a chemist, and it was absolutely clear. Of course, it was simple for us, simplified for us, but very good. And also, the connection with the diseases is important. Now there is a lot of effort to work on genetic disease. They are rare, but they are lessons for understanding also more diffuse diseases, so very important. And also, some of these diseases are really terrible, and I think that we have to do something to help these people to get better.
Yeah, yes, but to be honest, I have to repeat that our work, I think, that can only produce an insight, physical insight. Most of the work should be done by other scientists, like you, for example. I can only provide you with a very fascinating interpretation. Probably in the future, there might be some people who will be able of find an actual connection with this apparently far physical insight with the real disease. At the moment, I don't want to say that we can provide a very strong support to the people working in genetic disease. In my opinion, this is what I think about myself.
Yeah, but this is the spirit of multidisciplinarity,
yeah.
which is also the spirit of the TMS.
Yes.
Thank you.
Thank you.
Okay, thank you to Massimiliano. Is there any other question? So I think that, yeah, we have covered many questions. It was interesting, I think, just a note to Massimiliano say that it's important to understand what is the effect of a mutation. That this mutation can be also very far away from the active site. And this it's interesting, I think, because there are examples where this transfer of this mechanism of transfer of information from one side to the other can actually be quite different, as you might have experienced. So you might be.
Sorry, I lost the last part of your. I lost.
I mean, the transfer of the changes upon mutations that can be transferred through a long pathway to the active site to affect the active site. And this can be mechanical, can be dynamical, so it's quite a difficult effect to catch, but with molecular simulation, you can actually describe and understand deeper this the role also of why one mutation so far away can affect.
Yeah. I only want to leave you with this message, which is important, in my opinion. That is a phenomenon which takes place in very different time scale, very different time scale, might be strongly connected. This is quite unexpected from my point of view, and this is probably the most fascinating aspect of the story. So very slow motion do affect very fast processes like an electron transfer, and it's quite strange for me, but it seems that this is what is happening in the natural, at least in the natural molecules. In this case, of course. Okay.
Thank you very much. So
thank you.
There are no more questions, so I think yeah. Thank you.
Okay.
Thank you for
Thank you to everybody, and I hope it was interesting for you. Bye.
Thank you, Professor. Bye.